Okay, well, thank you everyone. I've been very much enjoying the conference so far. I think we had a very interesting talks during the yesterday, today, and Chris just did a really good job right just up front of my talk to actually line up all the difficulties that we're facing, and it's not just us, it's everyone. We have those still up front of us, and we want to do better or actually smarter to go around them, or actually introduce something that could help us to get into the biology of things. As you know, I have the HFX orbit trap with dual ultimate 3000 nanopump. I'm using pharmafluidics columns for all the work that I am going to describe today. And as you can see in the picture, actually not sure if you see my pointer. Here's my laser pointer. This is the basically setup I have. There is a 2 port valves. I'm using trapping columns from pharmafluidics. I'm using analytical columns, the old type of analytical columns, 50 centimeter one. And all the data I'm going to show is from 2019 data. I didn't do any single work on 2020 yet. And the column 1 was, at that time, was about 2 months old, and the column 2 was 9 months old. That's basically end of the column age that we have here, if you are dealing with those columns properly. And I think that's basically what the setup we have. Here, I would like to start from the old data that you already seen once, and then basically I kind of showed the last time, but I think it's very important. I very much agree with Nikolai that somebody who would actually reanalyze the data, and we get a support from the different techniques, since our single cell proteomics is kind of very young technique and is not necessarily, we can say 100% we're sure that it's what we show is truth. We always want to have something that actually confirm our findings. Okay, this is the data that I showed the last conference, where we have the microglia from the very old and venereal mice. That's a collaboration with the David Sinclair lab. And then just the extension of this study was that we used the activated microglia. As you can see, the FACS sorting, we start from this type of cell, then we have single cells, and we actually do 1 type of antibody, then another 2 type of antibody, to pick only the amount of the very small amount of cells that actually do relate to the activated microglia according to all these classical type of analysis. And we do have analysis of those from the young and old mice, and just we want to see the analysis. This slide you already seen probably from the last presentation I had. We have the 396 well plates. All we do is a 96 plate. It's been done with the scope MS, classical scope MS, was 90 minutes run. It was about the early 2019 data that we show that very well separation. And why I use this type of example, that we actually want to show that it's actually quality data, and this data is actually supporting something that we would like to bring to the field of biology, that single cell proteomics can do very interesting things. I just want to make sure that everyone understand that, you know, we have to be very carefully with the amount of things we impute in. We have to have a quality data. This is representation of this particular data. It's usually better than we have an average for a large project, but here's this, for example, plate 13 was this separate runs, where you can see the channels, and it's actually in the percentage how much we have a missing values of each sample. And then missing values are something that is very important, because that's what you're going to deal with. You cannot leave them as zeros, and in EA you have to impute them. And as much as you impute, as much you're getting away from the real biology. This is Israel. He did analysis of the data generally, and what we have seen, this is kind of pretty complicated plot. We have here some canonical markers for the activated microglia, and we found them. The black ones is the young, and red ones is the old mice, and as you can see, they're pretty much similar. There is not much we can see in the canonical things. And then we have found some markers for that have been upregulated and then downregulated in the single cells. But we actually found to be there is more interest in them, not just upregulated and been downregulated. They actually been bimodal distribution. There are some who actually up, some are down, and that's what brought us the idea we do really need to understand what we actually have here and how we can actually interpret this, and looking at the other technique that would try to confirm our results, unless we can just do something else. We took our data, and this is actually, again, 3 separate plates, so 96 plates worth of data. York was the person behind the analysis for this particular type of analysis. We prefer similar over the UMAP or TSNE for the small samples amount, and usually we do have a small type of samples, and we do separate them by plate to actually do a control on the batch effect between plates. We do combine them later, and you will see in the published results, they're all combined. But I just wanted to make sure that similar show that we have 1, 2, 3, in my opinion, maybe a little bit, this split in 2 type of the clustering. And this clustering, actually, as you can see, the dots are the young type of cells, and the ROMs are actually the cells that belongs to the very old microglia mice. And as you can see, that they've been separated pretty much in the clusters, but the clusters been separated between young and old mice, which we do expect. And in this plate, we do have similar behavior. We have 4 main clusters here, but we have interest in here in the plate 15, where we do have a separation of the clusters, but here in the red cluster, we do have a young and old mice type of activated microglia that cluster together. That's why the proteome-wise been much more closer each to other than actually age separation. And there was a very interesting findings, which we actually try to confirm by other techniques. And what we can do, we actually use the CyTAC. We took the antibody for the those that we can find, for those that we find to be activated microglia markers that found by single cell proteomics, and those that are classical markers. And you can see the markers here being run on the CyTAC instrument. It's about 70,000 cells. Roughly, there is less young, more old. And you can see that classical markers sometimes do not show any much difference here, which is findings here that we have, especially these 2, have a drastic difference on the abundance on the cells that have been confirmed by totally different type of instruments. And this is CyTAC from Aurora. And thank you, Zachary, for doing this. The UMAP analysis on the old microglia and young microglia actually do show there's about 1, 2, 3, 5, 6 main clusters that do confirm our findings that it's actually clustering within the cell type. There are different cell types in each type of mice, and we do have more clusters because clearly we do have much more cells being used for this type of study. And so I think it's a very good example of something which is single cell proteomics being found, which is not shown up on the bulk proteomics, and then being confirmed but independently with different type of techniques that shows drastic difference upon those newly found biomarkers. Okay, as I mentioned to you, we do like 96 well plates for the sorting. There is an old type of analysis. We've done the TT11 plex. That's type of channels we're using, going this way. And we did notice this 128C wasn't very good ion either. And it was nicely confirmed by Chris today. It's very nice confirmation from a group. Now the TMT16 plex goes this way. As you can see, we did remove 128C because we don't like it, since it was quite a bit of decoisolation. And that's what our kind of current way of doing the 16 plex versus 11 plex on the 96 well format. This is the next year, but the challenges are same as I've been mentioning in the previous year. It's exactly the same slide. We do have isolation interference. We know about it. There's nothing wrong with it. We do have missing quant data. Sometimes they are quite a big one. We want to reduce them because that's actually quality of the data that we are going to analyze further down. And then proteomic dynamic range. It's not something which is unique to the single cell proteomics. It's general things for the proteomics, but it's of course severe in the single cell proteomics type of analysis because we do have quite a bit of the problem to have enough data. Again, this is the old type of the scope MS analysis where we've been in the 2019 year. we are working closely to increase the amount of the cells. This is classical analysis of the differentiation of the Jurkat T cells versus U19037 cells. As you can see, we do have quite a good separation of the single plate. That's a 78 cells, 92 cells, and then it's just going to be close to the full possible capacity of 96 well plate. We do grow in the amount of the proteins we've been detecting, and that's the amount of the peptides is based on, and the amount of the PSM it was based on. All the data been run on the Proteome Discoverer plus Bionic type of search analysis. What the problems are still here, we do grow in the amount of the cells, we do have a noise, and as you can see, the separation is pretty well separated, but it wasn't separated on the PC2 because PC1 component still have quite a bit of noise, and we did kind of know about that. So we need to kind of address these things further down to have a quality data that we can actually start judging on the results and try to unveil the biology from such measurements. That's why we kind of try to go further and think about what we can do further. And of course, the DIA was kind of the idea. Okay, with the DIA, we can see everything. If we can see everything, we can actually potentially could get more data, and it could be better. And that's probably the solution, which is wasn't really in our case because we do not have an instrument that would actually do DLA pretty well, and we don't have a kind of software for the DIA type of analysis. That's why we just took another approach. We're not going towards the DIA, but we still would like to go towards the deeper proteome and the quality proteome. That's why we just would like to address those questions. I came up with the new type of platform, was the called SCREEN, Single Cell Proteome Analysis Platform. It's been used basically in the several different pages here. We use the carrier channel cells. This is the old numbers, it's quite a large numbers, but you can take any type of number. As soon as you do anything you do to the single cell, absolutely identically, that is very important. Your carrier channel must be prepared exactly the same as the single cell. Then you go to the high pH fractionation, you do your separation on the high pH, you run them, you do the searches, you create a library, and then you just go to the target that's run. All these are real pictures from the preliminary building up library after the high pH fractionation, and these are real pictures from the single cell run after that. That's why library-based scope X, that's how we call now our MS2 type of acquisition, where we do not accumulate, do not detect the 126 channel for the carrier channel at all. We kind of exclude it. You can start from 126.5 or 127.5, depending upon do you want to include or not, do you want to include the negative control ions for 127. And which is actually leaked from the carrier channel. And we would like to kind of make sure that we are running instrument not faster but smarter, because we would like to keep the depth of the what we achieved so far with the scope, but we would like to in the same time get the quality, because there are problems which we need to overcome with the isolation and other type of the problems that do have actually address our quality. This is the first type of analysis we've done by SCREEN platform. Again, we just want to be sure that our sorting, fact sorting to the 96 well plate or any other type of sorting does not really rely on our way to make a separation. That's our typical Jurkat versus U19037 cell sorting 1 by 1, 2 by 2, and 4 by 4. I think I described several times this type of sorting procedures. This is still TMT11 plex type of analysis. As you can see, in any case of scenario, we've been able to separate the Jurkat cells versus U cells pretty well, especially 4 by 4 sorting gives us quite a bit of a separation. And each of those plates, we got more than 2,900 proteins with 1% FDR per each plate of such analysis. This is those 3 plates together. Again, SCREEN platform by library targeted acquisition. We've been doing more than 30,000 peptides targeted during the 90 minutes run. And altogether, 3 plates, if you search them together with 1% FDR on both peptide and protein level. Although on Bionic, it's tough to say what they do on the peptide level. It's at 1% of their level. And there is no match between runs feature was used for this analysis. This is straight data analysis with the platform directly to the PD and output from the PD and our own analysis. We would like to go from these classical approaches between differentiation of the cell and go towards something which is more useful towards the kind of pharma industry and close to the disease relationship. And then we apply the screen MS platform to our classical, again, to us classical analysis, where we treat the Jurkat cells with the drug called PMA. And this is drug that interacts with the protein kinase C. Here we have the, again, type of 4x4 sorting, where the first 4 channels in the 96 well plate been sorted with untreated type of samples, and then next 4 is sorted with treated samples. Here we sorted with 2x2, again, 2, 2, 2, 2, and then those have been analyzed together. Again, we have a pretty good separation on the Jurkat cells that we've been untreated versus the treated cells. That's a kind of our proof that we can still do our analysis on the new screen platform. Now the next piece was that we started with this Sonic C group last year, showed a little bit this data on the scope MS, where we have the peptides that's been specifically synthetic peptides that have been introduced in the carrier channel as transcription factors. And we kind of try to cover that region of the depth of the proteome to go what we can see in the deeper proteome and how we can deal with it to see something that is biologically interesting, but unfortunately does not have enough copies per cell to pop up without any kind of help. And that help was a targeted analysis with the synthetic peptide. We continue this type of analysis. We use this 95 peptides that actually was provided by Sonic C to us and do the 90-minute gradient. As you can see here, the pharmafluidics column provides us exceptional reproducibility of the retention time. And I would like to actually repeat that column 1 was about 2 months old. Column 2, at that point, it was about 9 months old on that instrument. And you can see that the quality of the sharpness of the peak is actually start to be degraded, but I want to be sure that, you know, we want to actually challenge the instrument and columns to the possibilities that we actually do still have a quantitative data out of such runs. And you can see reproducibility on 90 minutes of each synthetic peptides. Awesome. And that gives us an ability to do actually targeted proteomics, which actually do allow us to do a smart runs rather than fast runs, and actually gives us ability to accumulate ions more than we have to, do repeat runs if we have to for exactly the same peptide, and so on. Here we would like, I would like to represent how we jump into the area where we've never been before. It's just the analysis of the Jurkat cells that have been PMA treated, and it's a time course, meaning that some of the cells been run, again, it's a 4x4 separation on the 96 well plate, but this is the PMA treatment for the 30 minutes versus 4 hours. And we do have a separation based on the screen platform between these two treatments. In the 30 minutes versus 4 hours, there's no cells really actually yet dying, but there is a different pathway that been involved in the cell reaction between the two times, and that's why we can separate them based on the proteome changes. If you take this data and put in the similar type of analysis, you can see that very important things that actually shows a true digital drug response. That there are some cells that are clustering here for this type of things, and you can see most of them are 4-hour type of treatment. There's a very few for the 30 minutes, whereas these clusters, it's about 50-50 actually do have from both types, 30 minutes and 4-hour treatment. And, you know, these two clusters, clearly there's only 30 minutes, but they are already differently separated on the similar, showing that there are some proteome changes between those two treated cells, even up to 30 minutes treatment. The next type of analysis we did together with the Vimisha, who provided to us cells She is a collaborator from Microglia lab here at Harvard. Here we have the two type of cells, which call HL60 and MX2. And those two cells are actually pretty much same type of cells, a cancer cell type. The only difference between them that MX2, it's a leukemia cells that are resistant to the drug called doxorubicin. And we would like to see if we could pick up those cells again with no any other tricks, just using the screen platform for the deep proteome analysis, and actually put them again, make 4x4 and 2x2 type of separation on 96 well, sorting to the 96 well plate, and we can see that we can actually pretty well separate them, even though it's practically the same cell types. The only difference between them that one cells are actually resistant to the drug. Further down, we would like to see how we actually implement this towards the real drug treatments, and then we use the screen analysis for take those exactly cells and actually treat them with the drug A, and drug A can tell you that was the doxorubicin, exactly the drug which is the MX2 cells being resistant to. And this analysis done by Alexandria from our research computing group. And as you can see, the difference here been separation of the MX2 cells being treated with one drug versus the cocktail of the drugs here. And here again is 296 well plate. I do believe this has been sorted 4x4. This has been sorted 2x2, again to show that sorting doesn't really matter how we sort the cell. They just been separated pretty well on based on the types of drug response that we see upon the different single drug versus actually two drugs in this case scenario. That's why we can we feel pretty comfortable that we have a very interesting platform that can go into the deep into the single cell proteome. We can actually prove that we see a difference between cells that are very similar, and we can actually see the digital drug response, not only just treated and untreated cells, but actually upon the different time of the drug treatments. That was very encouraging, and actually it's more work needs to be done to go further, but I think this platform is the future for analysis on such kind of type of Orbitraps. Then around that time, 2019, we had a conversation with someone who actually told me that it is great that you're doing this proteomics type of analysis. It's always nice, but always at the same point, it's easy. It's a proteomics, and you have this team T trick that you use to get your IDs. That's why you always have your IDs, and proteomics in generally is very simple things to do because you have IDs. It would be much harder to do something with the metabolomics. And of course, I always was thinking about what to do towards the true analysis of the multiomics of the single cell. And again, it's the same instrument. The everything, our philosophy here in the lab, that everything we do needs to be repeatable anywhere else. There is no single piece out of here on the instrument that you cannot purchase. Everything is purchasable, and there's no things that cannot be done on specific things. Everything can be done on average type of lab. Again, it's absolutely the same instrument. There is no changes was done whatsoever to the instrument to prove that we can do actually some kind of metabolomics work exactly on the same setup as we're doing for the single cell work. That's why everything we will see from metabolomics part of the analysis is clearly been done on the C18 column, because we didn't change columns. That's why exactly I just would like to mention first before we go further that what we see, it's only for those metabolites that first of all stick to the C18 material, and then second, most importantly, elute from the C18 material. Again, no changes to buffer whatsoever. It's normal buffer A with 0.1% formic acid, and normal buffer B, which is water and acid neutral respectively. That's why clearly it's not ideal buffers for the metabolomics, but the whole idea was to see what is the same instrument can do for the single cell proteomics and the single cell metabolomics. Okay, I would like to thank you, Alexander Bochenko and Mark DeFore, to actually help us to plumb the system properly, that we can actually use 100% of the system, because for the metabolomics, it's very important to have a very short runs. And very short runs, it's something that is very important, because if you do one single metabolites with 90 minutes gradient, it's not going too far. That's why you need to have a short runs. Okay, metabolomics, single cell preparation procedure. What you do, you principle, don't need to do any other instruments. Exactly what you have to do, you need to have a very good fax sorter. It's important. Fax sorter is the key. You do have the same small formulatrix robot, thank you, is beautiful thing. And you do have to have a speed vac, which holds 96 well plate. That's pretty much it, what you need to do. Everything else is exactly pretty much convertible to the single cell proteomics prep analysis. And we do confirm that after that, we can actually still see the proteins after the extraction of the metabolites. A couple of rounds of the work. This is our gradient. This is 20 minutes gradient total was created for such work. As I said, it's very much interexchangeable towards one another instrument until one is running. A second sample's already been loaded to the trapping column, and as soon as the instrument stops at 20 minutes, actually the second run is starting right away. There is no delay. It's 97% use of the HFX orbit trap. And again, that's the beautiful way to run the ultimate pump. Again, to remind you, column 1 was at that time about 2-3 months old. That's the 5 amino agent angiotensin peptide mix that we're using for such kind of quality controls. And the column 2 was about 9 months old. As you can see, some degradation of the separation of those 5 peaks, the 4th and 5th are kind of clumped together, but reproducibility of the retention times, that's a key here for the metabolomics, because clearly you do need rely on the retention time on any metabolomics analysis. Tomas was a collaborator who was interested in metabolomics of the myeloma patient cells, and actually he provided cells to us. That's why we've been able to do such interesting analysis. Again, there is no tricks here. We do basically 96 well plate. First 2 we put as 0, just the water. Everything else we put as the single cell, and on the channel 12 we put 100 cells here. What the amount of how we set up the experiment, it was a plate which is we run column 1, column 2, column 1, column 2, and these were not treated. Then it's treated with a drug called BTZ, LBH, and lean. These drugs here. That's a typical myeloma drug, very well known, and we would like to see, can we do separate cells upon the drug treatments just based on the metabolomics analysis. As I said, there was quite a first step into the metabolomics analysis. I have not been doing metabolomics for around 10 years. I kind of look into the things where metabolomics stands. I would say not much change since I dropped it 10 years ago. The only things which was improved, it's dramatically improved, the quality of the instrumentation we have, and the community effort to create much more accessible databases. And the databases, that's a crucial point for us to do anything which is metabolomic, because we don't have, when we don't use any heavy standards here for this analysis. This is how the real data looks like. This is metabolomics analysis from our HFX orbit traps, 20 minutes runs. As I mentioned, this is no labeling here, single cell extracted metabolites been loaded on the C18 trapping column, been eluted from C18 trapping column to the C18 analytical column, and run in the both modes, positive and negative mode. It was quite tough to create a negative mode that is reproducible. That's how the data look like. In the end, we are running first the blanks, then after the blanks, we're running real zeros. After the real zeros, we're running single cells, and after the single cells, in the end, we're running 100 cells with MS2 to gain some identification from MS2 on the single cells, and then rely back them towards the single cell analysis. As I mentioned, this is the setup of the treatment. We have no treatment. We have a treatment A, treatment B, and treatment C, and we would like to see if we can actually do separate single cell based on metabolites and based on the analysis. I do agree with Nikolai that sharing data and actually do ask somebody else to search or analyze the data is very critical, very important step. That's why I don't think I can say that I'm absolute professional in the metabolomics analysis. That's why we've done our data analysis ourselves, and we did share it with somebody else who we think are professional in such analysis to confirm what our findings are. Here again, all search has been done by the Compound Discoverer version 3.1 from Thermo. It's a beautiful piece that they put together. After the Proteome Discoverer, the Compound Discoverer was the second program that they put together. It looks great. We did normalize each drug treatment to the blank runs. We removed compounds from 150 to 150, which is probably right now not sounds very well to somebody who is doing metabolites, because it's plenty and huge amount of metabolites in this area. But as I said, we're not professional. We decided to remove this area because it was quite a bit of noise there as well too. So I removed those. Clearly we lose some metabolites from this area. We use only the top 25% of the abundance for the peaks, and we only took top 25% of the CV of each type of the treatment and not treated. And actually top 25% of the CV was defined as a 25 lowest CV percentage across each of the 3 drugs, and the control was the range for the each drug being separately. Okay, what do we see? We have the plate 1, 96 well plate, and plate 2. We have column 1 and we have column 2. On the column 1, Compound Discoverer came out with the 13,302 compounds. That's what the Compound Discoverer call compound. Doesn't necessarily mean being identified, but it's been identified across all the runs and assigned as an individual peak. And then on the plate 1 is 96 well plate, column 2, we have the 9,731 compounds, the smallest amount of the compounds we detect in this study. And I have to say that this is compound being combined from the positive and negative runs. The Compound Discoverer put them together. That's why we do not have separation from the positive and negative type of runs. This is the PCA that we came up with. As you can see, there is a 3 type of treatments, and then these are not treated cells, versus here we have on the column 2, which was, again, I would like to repeat, this 9-month-old column, a very much dirty one, and we do have quite a kind of tough separation between not treated and LBH, which is very gentle treatment type of drug here, and these two are separated pretty well. Then we tried to improve a little bit extraction of metabolites, and for the plate 2, we improve our analysis. We came up with the 19,598 compounds being defined by Compound Discoverer. And on the plate 2, column 2, again, is the same column, no changes the columns. We just jumped from 9,000 to 1,265 compounds being identified on the metabolomics of the single cell. Again, separation is here now clearly pretty good to me. And now we've been able to separate as well these 2 problematic that we have not been able to separate before. As I said, we kind of cannot tell ourselves how good our result. That's why we would like to have someone else look at our data as well. We asked our collaborators from the Revive Med, Leila, she, thank you, accept this challenge and try to look at the results. That's their workflow. They took the raw data from us. They run through the XCMS, identify peaks, do normalization, do hierarchical clustering, and PCA analysis as well. And the all work was done by James, and they revealed the data to us back, and that's what they see. They have the identification, again, the same type of analysis of the data, plate 1, plate 2, column 1, column 2, and they do have a separation in positive and negative mode, and that's type of amount of the significant features, they call them features, versus the compound from the Compound Discoverer, the features that they could separate those things apart, and they have pretty much confirmed our separation between each type of cells, each type of treatments, and on the top of that, they kind of confirmed that they do have a separation on the treatments between each drug, not only drug on drug type of compound. And here we have the, in their opinion, the TTS p-value less than 0.001, how many features, what they call, being able to identify between each type of drug treatment. Again, this data from Revive Med, and we hope to publish it later for type of metabolomics of the of the single cell. Again, I would like to confirm that this is C18 column only. That's why clearly there is not full metabolome was analyzed. Going back to the single cell omics challenges, now we have proteomics, now we have shown metabolomics analysis of the single cell. Where the challenges are, they stay pretty much there. We would like to be smarter, not faster on the MS instruments, because our MS instruments are already fast enough, but they are doing majority of time complete garbage work. We need to make sure that they do exactly what we need them to do, to look at the peptides that are eluting. We need to point at those peptides and let them to take as many microseconds they need to do to collect the quality data. What else will we need to do? We still want to go towards the RNA-seq type of analysis. Yes, we would like to go towards 10K depths of the proteome analysis, because that's achievable, it's doable, we just need to get there. And of course, that's a challenge, and we are as a community should go towards that challenge. Now there is that actually metabolome added to the picture, that we do want to go towards the metabolome, deep metabolome analysis of the single cell. Reproducibility of the sample prep, the QC of the studies is very important. I absolutely agree with Nikolai, with the community, that all the studies we reveal should be shared on the Pride or any other type of sources where the people can come back and just relook at the data, maybe came up with the better type of analysis of the data, and actually the quality of the data is very important. Now the amount of cells per analysis, it's it was very important for the proteomics, because in proteomics we do have only 12 cells from out of 16 plex type of things. It is much more severe with the metabolomics, because you cannot afford any more 90 minutes runs, because it's a single cell, you do not have any label, and you do one by one. It's a label free. It's basically will take quite a bit of instrument time to get to something usable. That's why short gradients is targeted gradients. That's the way to go, and that's, I think, where the next steps of the community should be concentrated on the quality of the preparation of the samples. Because in my opinion, the quality of the runs and quality of the instruments repeats and quality of the columns and other pieces are much less contribute to the quality of the set compared to the preparation of the single cell. That's where the largest error bar is coming from. The new frontiers for single cell omics, as I said, the current state of my lab for 2019, I didn't do anything for this year. Screening mass was developed with the coverage of more than 2,900 proteins from a single plate. If you do TMT6, 16-plex analysis, you're analyzing 12 samples per run, and it's about 1,000 cells per week of instrument time if you do the double pump. Single cell metabolomics based on the C18 columns achieved. I hope I convince you that we've done some. And some future perspectives for 2020 plus. We need to create more reproducible single cell sample preparation procedures that will contribute to the quality control. Make faster analysis for the larger projects that we can compare to the single cell RNA-seq technologies. Again, it's not the question that we need to draw those beautiful pictures that you've seen on my first slide. I think that's pretty much our current art that John Yates represented yesterday beautifully. I think that's currently state of the art, what is art, but we want to do towards science, and towards science we need more cells to get to those rare amounts. And if we want to have a rare amounts, we need to have more cells. That's why we need to do smarter and shorter runs. And we need to build further technologies toward true multi-omics analysis of the single cells, because that's our first step towards metabolomics, and I think community will follow and go through it. My own things, I would like really to see what the screen platform plus stream analysis would show. There's a very interesting pictures was shown yesterday, and I'm really looking forward to reanalyze our data with the stream analysis and see how they will look like. At this point, I would like to say that I think, you know, I showed previously some first airplanes from the brothers Wright that didn't just barely take off. I think at this point, I would like to say that we are in the air. We took off, it's there. I think single cell proteomics now is a realistic thing. And I would like to thank you all my collaborators from all these groups and ReviveMed, of course, as well as the clearly Harvard University for supporting these things, and my colleagues from the research computing team for building up tools for single cell analysis and single cell visualization data, which is a very important piece of our work. And I have to say that I reveal I'm a scientific advisor to the Merck. And I do have the last, but not the least, slide to show. I've been invited to be a guest editor to the specific single cell omics molecular omics journal issue that we organize in. And I would like to invite all our speakers from this conference to participate in this special issue, or anyone from the audience who has data for true single cell proteomics type of data to please submit data to this coming issue. You can just look from this web page and follow the instructions. And I would be glad to take any questions if you have. Thank you for your attention.